Ladies and gentlemen, it's my pleasure to welcome you to this event of the College of Engineering Distinguished Lecture Series. We began this in 2009 in response to the National Academy's issuing of the grand challenges for the engineering field. Very timely proclamation, and engaged many of us around the nation to concentrate on some things that were very, very important. On the surface, these are probably more technical themes to work on, energy from fusion, managing the nitrogen cycle, clean water, and so on. But what bound these together was a very, very important theme, providing a new face for engineering. The fact that it matters and improves quality of life on those things that are most important to us, and they are sustainability, health, security, and joy. And these grand challenges all relate to these important imperatives. And as we think about the challenges ahead, it is important to recognize that none of these challenges can be addressed by one single discipline. It requires all of us to work together. And for an educational environment like us, that we want to engage our students who are driven by passion, curiosity, and engagements and dreams, to provide them with an environment that helps them with their learning and pushes them to think more and more critically. And this lecture is just another part of what we would like to provide to our students and help them see what's most important to them. These lecture series are organized by the Dean's Advisory Council. We are thankful for all of their work. A subcommittee of this council consisting of Nick Ceramic, Susan Fort O'Neill, Michael Nigley, Kent Peterson, and Matt Petreem have organized this event. Please give them a round of applause for their hard work. Several other members of the Dean's Advisory Council are present, and I would like to recognize them. Dan Craig, Chris Hernandez, Saeed Hilal, Mel Marks, I don't know if Mel made it or not, and Al Morrow, thank you for your participation. This event is made possible by the sponsorship of a number of our friends, Applied Medical, San Diego Gas and Electric, P2S Engineering, Port of Long Beach, Fort Design, and TechnoLink Association. Thank you for your continued support. We do have some special guests. Andrea Taylor, Vice President for University Relations and Development at Cal State Long Beach. Thank you for your participation. And Councilman James Johnson. And I'm particularly pleased that our Provost, Dr. David Dowell, is here with us to welcome you on behalf of the university. Dr. Dowell is considered the force behind all of our student success initiatives, and we owe much of our success in this area to his relentless, tireless work on this campus. As a former chair of psychology, he has, and a professor of psychology, he has a particular interest in this topic. Please welcome Provost David Dowell.
Thank you, Forzan. On behalf of the faculty, staff, and students of the university, and especially of the Division of Academic Affairs, I welcome you to the campus and to this event. You could hardly have chosen a topic of greater interest to me. As a scientific psychologist myself and always fascinated by the workings of the brain, this is really going to be an exciting panel with a very distinguished group. You know, philosophers call the problem of consciousness the hard problem. But if there's one lesson to be learned from science since Darwin, is that dazzling complexity can emerge from the simplest of processes. And I suspect that some of what we're going to hear today is about how complexity can emerge from the simple processes that underlie it. So I'm very excited to hear what's forthcoming, and thank you for being with us. And thank you to Forzon and to the committee and the college for organizing such an exciting event that contributes to the intellectual climate of our campus. Thank you.
Thank you, Dr. Dowell. And our president, Dr. Don Parra, runs in 20 different directions in any given day. I'm glad I saw him at another event at lunchtime. Unfortunately, he couldn't be here. And we heard a very, very kind message from the incoming president, Dr. Jane Connolly. She was gracious to send me a note. And I'd like to share with you what she expressed in her note. She says, I wish I could be at tonight's distinguished lecture series on engineering the brain. Neuroscience is a passion of mine. And she goes on to thank the college and its advisory council for putting together this event that really brings the community and the university together in such a nice way. And on to the main event of today, reverse engineering of the brain. It's my pleasure to introduce the moderator for tonight, Mr. Nick Ceramic. He's a senior project leader in systems engineering for the Aerospace Corporation, where he has worked for over 30 years, primarily on military satellite systems. He is an American Institute of Aeronautics and Astronautics Associate Fellow and a Computer Systems Technical Committee member. I'm really proud to say that Mr. Ceramic is a distinguished alum for the College of Engineering of Cal State Long Beach, and he has earned not one but 2 degrees, bachelor's and master's degrees from us in electrical engineering. He's a member of the Dean's Advisory Council, and he chairs the subcommittee for organizing these lecture series. Please welcome Mr. Nick Ceramic.
Thank you, Dean. I wanted to thank everybody for being here. I see my sister-in-law's here. We actually got into an interesting conversation about this topic about less than a week ago. So I think she's educating me more on this. She actually works here at Cal State. I'm really excited to have you all. I'd like to welcome you again. And, you know, Hilda. I'd really like to welcome everybody. What an exciting topic. I think this is going to be one of our most exciting topics. We've had some real great topics for those of you who have been here. And we got to actually see video of open heart surgery, actually watch the surgery. We've had some excellent presentations, good discussions here. And what I want to do is I'm going to give a short lead in to this just to kind of set the stage. But afterwards, we want to leave enough time for questions. I think there are going to be a lot of questions here. I have some real good questions in case there aren't. But I think there are going to be a lot of good questions. So I really want to get this thing moving. Let's see if I can get this done then. Okay. It's really interesting that we have this topic, and we had been really working on this topic for probably about a year now or so, talking about the topic. President Barack Obama actually just signed this month a new initiative that's actually been in the works for a number of years that really wants to look at mapping the brain. It's called Brain Activity Map Project. And it's going to be about $100 million a year. Number of scientists who are working this subject have been the developers of really the subject and trying to get the money put into it, have really wanted to have this thing go multiple years. They really want to have this thing go out there, $100 million a year if they can get that. I mean, it's tremendous money to go into such an important topic. One thing about it is in looking up this and trying to learn about it, I'm an engineer, do satellite systems. I don't do brains, but it's been a lot of fun trying to understand this topic. And currently, the way I understand it, and and you guys will probably correct me as we go along, that activity can actually be the neuron, which is the brain nerve, that are actually interconnected, and I'll talk a little bit more about that later, that scientists now can actually monitor single neurons, okay, using electrodes. They can also monitor the entire brain or parts of the brain using functional MRIs, and there's other equipment, I guess, that's available. Hopefully we'll hear more about some of these things. But there's a whole area in between going from one SAMP electrode, and the scientists really, they want to get to where they can do maybe 1 million, 10 million, 100 million neurons at a time and really understand the brain. So that's really some basic questions that they really have out there is, what does that middle ground really look like? Okay, you go from one and being able to really stimulate it to, and I'll show the numbers, it's many billions of neurons, that comprise the brain. So, it's really an interesting topic, and I think hopefully this money will start getting us there. Okay, I wanted to put this up. This was actually from the Allen Institute, and I want to thank Dean for this picture. I thought it was great because as an engineer, I can relate to this. When I do computer systems, or anybody does computer systems, the hardest part today, because computer systems are comprised of hundreds of millions of gates now, transistors, and to interconnect that is probably the most difficult program, or part of building a computer today. That's where all the real problems are. So understanding interconnections, I think I can relate to it, because of a picture like this. How do the neurons connect to each other? How do they talk to each other? How do they communicate with each other? So for people to start really understanding it, I think from my standpoint, down the road as we understand more of the brain and the interconnecting and how everything talks to each other and communicates with each other in the brain, I think it'll really help us down the road in developing computer systems. We may take leaps once we start understanding the brain. Okay, I wanted to put this up. This was actually kind of interesting picture that when you talk about they can stimulate one neuron now, they can look at the brain through with different pieces of equipment, and stimulate different parts of the brain. But when you go from one neuron to 86 billion neurons, okay, there's a big leap in there. So I thought this picture is very interesting. There's a lot of research being done on worms, on mouses. I think one of our speakers is going to talk to some of that. But you really want to start small, trying to understand it. But to get up to the human brain, I think it's going to be very, very interesting. So I want to get off the stage, get our speakers up here. You're going to hear some very, very interesting different areas of research that these people are working in, and different things that are going on out there. I thought this topic's just fascinating, and we got 3 just excellent speakers. One thing I just want to mention was outside we all got these books that have their bios in them. I started a few times ago, a few of these lecture series ago, that I didn't want to take the time to read each person's bio. I can really appreciate it. I'll tell you, they're tremendous, have tremendous, bios in here. I really encourage everybody to read their bios, but I don't want to take the time to read your bio. It's a very distinguished panel. So what I want to do then is just start out. I'd like to introduce Dr. Mario Mendez. He's Director of Neurobehavior at the VA Hospital, Greater Los Angeles. Okay? Oh, no. Do you want to use that? Okay.
This thing on?
I think you should be able to get. Okay, you're on.
thank you very much for the invitation, Dean Golshani and colleagues. It's wonderful to be down here. The weather's beautiful here. And I'm from LA, so, you know, I asked, when I was invited to do this, I didn't know, and I still don't know that much about what reverse engineering really means, but one of my sons is a grad student in mechanical engineering, and I called him up and I asked him, can you help me on this? Because I'm going to be talking to engineers. And he spent a half an hour on the phone with me explaining exactly what I need to discuss with you. And I'm sorry to tell you that I didn't understand a word he said. It was all differential equation. So I'm going to revert to the kind of research that we do. And the first task is, of course, to move forward on this thing. So in thinking about reverse engineering, probably, at least my way of looking at it is it's a way of simulating or modeling the brain so that you can analyze the brain and use brain function to not only understand normal neurologic function, but also neurologic disease, which is what I deal in, and also development of mind-like intelligence machines. And we don't have one here, do we? We're going to look at the brain and model the brain in order to understand the brain and understand disease. So it's a process of analysis and synthesis. Now, I'm not going to talk about creating a silicon-based model of the brain. And so what I'm going to talk about is a simpler way of modeling brain function, and that is looking at neural networks and computational models in order to understand dysfunction. And I'm going to use two examples of the kind of research that we're doing. Just a few brain facts. First of all, the brain is, I think about it as a 3-pound sponge just floating around in a bunch of liquid, tethered at several points. And a very fragile thing, but at least it's in a hard box. It's composed mostly of water. And interestingly enough, we have more neurons in the Los Angeles area than in the LA city area north of here. 100 billion neurons than your 86 billion neurons, Nick. And I don't know why that is, but I think we're born with about 120 billion neurons, and actually they're pruned like crazy down to 100 or less, depending on where you go to school. Glial cells are 10 to 50 times that. The surface area is about 2.5 feet. The thickness is only a few millimeters, and there's 6 cortical layers, and I particularly want to point out that about 41% of the human brain is the frontal lobes, the crown jewel of human, or the crown jewel of evolution, if you put humans on the apex of that process. So we have different interacting layers, and these layers basically allow us to form neural networks, or to think about both a lot of hidden layers, top-down, bottom-up processes. And this interacting mesh is what we're trying to decipher in various systems, whether it's vision or visual perceptual processes or language, memory, executive abilities, conceptual abilities, whatever process that you happen to be interested in. Now, I have a colleague up at Stanford who initially used resting state fMRI to map out the resting network, or the default neural network, which we've been studying in conditions such as Alzheimer's disease. But what I want to talk to you about is 2 areas of research in the short time that I have with you. One is frontotemporal dementia, which is a lot of the research that I have been doing. And that has to do with that mysterious 41% of the brain, which is the frontal lobes, which, as you can see, was an evolutionary development. Now, one of the things that the frontal lobes does is it holds information online. And the more information, more bits of information that you can hold online, the more you can interrelate them. And interrelating information allows you to analyze information and relate it in various increasing levels of complexity. And of course, this was worked out at UCLA by Joaquin Fuster in the macaque, showing that indeed you have increased neural activity when the monkey is just holding that information during a delay period. So that this process of what we call working memory, of maintaining and manipulating bits of information, is really critical to the frontal lobe function and the ability to handle increasing levels of complexity. And you can model that on a computer, and we can apply that to a lot of our patients, and hopefully to develop technological ways of improving their ability to hold information online. Whether it eventually is a process of inserting microchips in the frontal lobes or whatever, this is where we're heading. Now, a couple of areas of research that we do. One relates to modeling the social brain. And this is one of my passions. I've been trying to work on that for a long time. And this, I believe, is the world's most important neurologic patient. Many of you know Phineas Gage. Phineas Gage was a straight, very traditional foreman on the Vermont and Rutledge Railroad when on September 13th, 1848, that tamponading rod went right through the medial aspects of his frontal lobes and gave us a big clue that that's where a lot of social behavior is modeled, and that's where a lot of social behavior is mediated. And this is a disease model that we're using. This is a patient with frontotemporal dementia where you can see the demarcated line of atrophy involving the frontal lobes. So in these patients, we can look at the models that we create of the social brain and how social behavior is situated. This is one of our studies using diffusion tensor imaging and looking at the white matter tracks. Actually, it turns out that they're pretty much wiped out. As you can see, there's no frontal lobes in this study. This is another study from one of my colleagues looking at the actual networks involved. And most recently, we've been modeling the social brain network and its connections to more posterior regions of the brain using not only diffusion tensor imaging, but a form of mathematical assessment, kind of a complicated hierarchical cluster analysis where we can actually look at where the hubs of these centers are. Centers for empathy, actually centers for moral behavior, centers for theory of mind, and other basic aspects of social cognition. Okay, so the last part I want to talk to you about is more recent work that we've done with head injury. Now, the head injury, as you know, there's a lot of work now with chronic traumatic encephalopathy, but many years ago we started out looking at this in boxers. And of course, the frontal lobes are very susceptible to a blow like this. And I remember a study done a long time ago in the UK where they actually measured what a heavyweight blow to the face, the power of that, but using strain gauges and all of that. And translating it into the English system from the metric system, it's like getting hit in the face with a 1.7-pound mallet at 100 miles an hour. So if you think that doesn't do anything to the underlying 3-pound sponge floating in a vat of liquid and tethered at various points, then you just, you know, I'm not making any headway here. Now, what interested us in the VA side is the fact that many of our veterans are coming back with symptoms of traumatic brain injury, and usually mild traumatic brain injury, but from exposure to blast forces, changes in barotrauma, changes in environmental pressure, rapid changes with without the usual blunt trauma that we're used to studying. In fact, in the most recent conflicts, we've had ever-increasing percentages of veterans who have been exposed, whose primary exposure has been to explosive devices, emitting these massive changes in atmospheric pressure. And I'm not talking about secondary, tertiary, or quaternary exposures such as to, associated projectiles being body displacement, chemical exposure from the blast itself, but just the primary blast forces. And those of you who know about this know obviously that it is, when you analyze out the basic curve, it's discussed as the Friedlander effect, or the Friedlander curve, where you have a huge, like in B here, a huge increase in milliseconds or less of exposure to increased pressure with afterwards a fall below atmospheric pressure. So if you take this 3-pound sponge, then you're thinking about compressing it and then expanding it, and it's just not good for the neurons. Now, this is not my work, so it's cited down there just to be clear, but this also affects those axons that we've been discussing. They are damaged, and there are also deformation forces in the brain that are distributed. So some parts are compressed and some parts are enlarged. This is not my work either. It's referenced. And we also have areas that we've learned are particularly affected, such as there are red arrows there in the center projection, showing that wherever there is a connection between the brain tissue and the meninges and nerves that tether it, you have particular damage. And all of this can happen when you're exposed to these environmental forces. We did try to contribute to some of the development of this in terms of the Kevlar helmets. And one of the ideas was to not only increase the padding within the Kevlar helmet, but to create various protective layers like a Russian nesting doll that would absorb a lot of the environmental forces and environmental pressure. And of course, we've been worrying about the frontal lobes, and there's no frontal protection for that part of the brain. So I think I'm within my time limit, but right now we're trying to look at head trauma and its relationship to dementia and its relationship to the development of some of the neuropathologic changes that we see in dementia. So I think that's it.
Great. Thank you. Got it? Okay. Thank you, Dr. Mendez. Some of this work that he's doing, in fact, all the work he's doing, I think is just, it's with dementia, with Alzheimer's, all these things happening to us seniors as we get older, I hopefully this goes a long ways, and I really hope, and in fact I was going to ask the question if you guys get up there, but just to mention that I'm really hoping that this money that the President's putting into this initiative, really advances the state of the art, the understanding, and what can be done about some of these things. So, okay, enough for me. Next, I'd like to introduce Dr. Roy Hamilton. He's assistant professor of neurology at the University of Pennsylvania, and he's come out here for this. We really appreciate it. Thank you.
So let me start by saying what a pleasure and an honor it is to speak here today. I realized that coming from University of Pennsylvania, I might seem like an out-of-towner, so let me just burnish my Long Beach credentials for a moment. So I was born and raised here, actually just a few blocks away. My mother graduated here, got her master's degree in linguistics, so I guess technically that makes this my alma mater. And so this is really a place that I consider to be where my family took its first steps towards academia. So thank you for allowing me to come home and tell you what I've been doing with the last 15, 20 years of my life. All right, so I want to talk to you about what I've been working on, which is neuroplasticity, neural repair, and non-invasive brain stimulation. These are my disclosures. And to kick things off, I know this is supposed to be a talk about what's new and modern and maybe even futuristic, but I'm going to start with something that's very old, and that is this. So some of you might recognize this. This is a phrenological map. It's actually the cover page of the American Phrenological Journal. You can see the date right there, March 1848. And for those of you who aren't familiar with the pseudoscience of phrenology from the 19th century, it was this idea that different parts of the brain were very specifically tied to different aspects of mental ability, but also to personality and character. So closely tied that, in fact, you could determine a person's character by the size of those areas and correspondingly the size of the overlapping or the overlaying skull, right? So if you sort of felt a person's bumps, lumps, and prominences, you could tell all about their personalities and what they were going to be like. Obviously, we do not hold to this. This is not the current standard of practice in neurology. However, I do think that it is emblematic of a certain kind of thinking about the brain that persists to this day. Right? And that thinking is that the functions of the brain are relatively fixed, that there are certain spots in the brain, the spot X does Y, and that's what it does. Right? And that kind of thinking leads to the idea that the plasticity of the brain is limited, that if you have injury to some region and that region is meant to instantiate some kind of mental operation or function, well, too bad for that operation, right? That the plasticity and potential for recovery is highly limited. And so what I want to do today is give you two short vignettes from the work that I've done over a number of years that suggests that these three ideas are largely untrue. And so the two stories I want to tell you about today have to do with the sensory abilities of individuals who have blindness and also the recovery from aphasia, where aphasia is an acquired language deficit. So recovery from aphasia in patients who have had strokes. So why blindness? Well, at least one reason that we would study this or want to study it is that we know that blind individuals have remarkable compensatory sensory abilities compared to sighted individuals outside the domain of sight. So this is some work done a number of years ago that I was involved in with my original first mentor during my training, Alvaro Pasqualeone, in Boston. And the point here is to demonstrate that blind individuals, when tested using these gratings up here on the top right with different spatial frequencies of patterns, when tested using those gratings, blind individuals are exquisitely sensitive to the orientation of those gratings along their fingers compared to sighted individuals. And you can bring that out even further if you touch them on the fingers that they mainly use to read. Most blind individuals have a sort of main reading finger. So if you touch that main reading finger, it's exquisitely sensitive compared to other fingers with respect to tactile discrimination ability. If you then take those patients and you put them in an fMRI scanner, functional magnetic resonance imaging scanner, and you touch them on those fingers, what you get is differential activation of the brain depending on which finger you are touching. And when you touch that reading finger, that exquisitely sensitive finger, what you get is preferential activation in the back of the brain. So down there, that bottom figure, right? An area of the brain that individuals who are sighted use for vision, implying that at least in part, the exquisite sensory compensatory ability of these individuals is mediated by their ability to recruit the area of the brain that you and I, or other sighted individuals at least, use for vision in the service of touch. This is further borne out by at least one case. I was fortunate to be able to report on a patient who herself was unfortunate in that she suffered from a stroke that affected that region of the brain. Right? And so it affected both sides of the brain, and this person was congenitally blind. She was actually a blind Braille reader. She was a proofreader for Braille. That was her job. And what she found was that after she had that stroke, since she didn't have vision to begin with, nothing happened to her vision, but she could no longer read Braille. Right? And again, just to remind you, that sort of is the flip side, the corollary of imaging findings that demonstrate that when individuals are reading Braille, they activate that posterior region of the brain. So we asked ourselves whether or not we could potentially experiment, maybe manipulate this process experimentally to learn more about it. Well, in order to do that, we would need to have some type of technology, some type of tool that enables us to focally manipulate brain activity in adult human beings from the outside. And that's the role of the main workhorse of my laboratory, transcranial magnetic stimulation. So for those of you who aren't familiar with TMS, it's based on the Faraday principle. So that idea is that if you have a coil of wire, I'm talking to a bunch of engineers, you guys know this. So I have a coil of wire and I pass a bunch of electricity through it. Right? When I pass electricity through that coil of wire, that creates an electromagnet, right? And with a fluxing magnetic field, if you have a second conductive body in the presence of that fluxing magnetic field, you will generate current in that second body. Right? In this case, in the case of TMS, that second body is the neurons of the cortex. Right? And so that allows us to depolarize cortical neurons in a focal fashion. It's non-invasive, it's reversible, and importantly for the purposes of our experiments and for therapies, it allows us to disrupt and manipulate brain activity. So, getting back to our blind individuals, when we put that coil over the back of the head, over the region of the brain that these blind individuals seem to be using to enhance their touch abilities, and we compare that to when we put it over the somatosensory cortex, the part of the brain that we commonly associate with touch ability, what we find is that when we place it over that somatosensory cortex and present Braille to these individuals, they say, "Oh, did you even touch me? I didn't feel anything." When you zap that touch center of the brain, but when you move it back to that center of the brain that sighted people use for vision, that these individuals appear to be recruiting, what they say is something different. They say, "Oh, I felt you touch me. I just couldn't figure out what it was. I couldn't make it out. It's indistinct to me." So somehow, these individuals are using that region of the brain, recruiting that region of the brain for the purposes of perception, and sort of the finer perception of these elements. Well, then we asked ourselves, well, is it the case that this is because there are connections from, whatever systems mediate touch information to that back area of the brain de novo in these individuals? Is it the case that blind individuals make these connections and sighted individuals don't have them at all? Well, in order to address that, we brought in some sighted people. We brought them to the hospital, blindfolded them, kept them in the hospital for a week, made them train on Braille tasks, all sorts of tactile training, continuously. As you can imagine, we paid them handsomely. And then tested their touch ability, their ability to discriminate Braille, scanned them to see what areas of the brain activated, and then also saw whether or not TMS to that region I was telling you about disrupted their ability. And lo and behold, they got better at their Braille discrimination over time, and that improvement correlated with activation of that back area of the brain I was telling you about, that occipital cortex. And when we stimulated that region of the brain, it created errors in their Braille reading back to their baseline. Right? And so the idea here is that not only in blind individuals, but also in sighted individuals, we are capable, the brain is capable of dramatic plasticity with respect to its functional use of the brain for different activities in response to demand. Right? This doesn't normally happen in sighted individuals. It's when they're deafferented. It's when they need that sort of extra processing with respect to tactile ability that this seems to come to the fore. So let's switch to our other vignette now. Right? Keeping with this theme of the brain that is capable of dramatic plasticity in the setting of need. I want to switch to individuals who've had strokes and aphasia. So just to set the stage a little bit, most of the time aphasia is associated with damage to the left hemisphere of the brain. We typically think of the left hemisphere as being predominantly dominant for language processing. Right? However, if you scan individuals who have aphasia when they're in the chronic stage, well, first of all, it tends to get somewhat better over time. And if you scan them trying to do language tasks, what you see is that rather than activating just a left hemisphere network, as normal individuals would, that's what's in the blue, individuals with aphasia will activate a bilateral network. They'll activate areas in the left hemisphere, sort of around the areas that were damaged. They'll also activate the right hemisphere. Right? There's a much more broad distributed network of areas that seems to be doing something in the service of language for these individuals after they've been injured in the area that normally does it. Well, we looked at this a little bit closer. We sort of developed algorithms and ways of looking at our imaging data. And by the way, you cannot read that font. I understand that. Okay? You're not meant to. I'll point out what's important in a second. So we broke it down into an algorithm looking at which areas of the brain in that distributed network of reorganized centers in these aphasic individuals, which areas seem to actually be functionally compensating or doing something helpful with respect to language processing, and which areas seem, I don't know, just activated but maybe irrelevant or maybe even deleterious. And what we found was that for the most part, most of the areas that we found in this distributed network are actually helpful, compensatory, both in the left hemisphere and the right hemisphere. We actually found one nefarious character, though, right here. So there's what the arrows are, and that's where it falls on our algorithm there. That's a center in the right inferior frontal gyrus, a region known as the pars triangularis, or the inferior portion of that region. It just seemed to be activated in ways that did not seem to be helpful with respect to language recovery. Well, it turns out that if you take TMS, right, and you start roaming around the right hemisphere in these individuals with stroke and aphasia, disrupting transiently, thankfully transiently, disrupting different areas, and you get to that pars triangularis, that one site in the right hemisphere, their ability to name things, this is sort of a measure of some language ability in these patients with aphasia, will transiently improve. Right? So we took this center that we thought was noisy or otherwise not helpful in the network, and we suppressed it, and in response to that, it seems like their ability of these aphasic individuals at least temporarily improves. Well, so then we took that site, after finding an area that is most responsive for each individual, and we stimulated that for 10 days in a row. We brought them in and just stimulated 20 minutes of TMS just to that one spot of the brain, and then followed them up, say, months, weeks and months afterwards to see how their language ability fared. And what we found was that, indeed, the ability to name things, so this is action naming, animal naming, tool and implement naming, improved 2 months after stimulation, 6 months after stimulation, as well as overall fluency. We were recruiting patients who had what's called non-fluent aphasia, so their main problem was producing language. And so what we found that when they were trying to describe a complex scene like the one depicted on the upper right, that their ability to produce different parts of speech, nouns and verbs and other parts of speech, as well as the number of words they used to generate to describe the scene above there, increased, improved as a function of having received TMS. But the thing about TMS, I wish I had time to show you exactly how we do it and how big the device is and how you have to stay still to do it. It sure would be nice if we had an easier way to stimulate the brain, sort of a more convenient way to do it. And so that sort of brings us to the other workhorse of my laboratory, which is transcranial direct current stimulation. So if the basic idea behind TDC or behind TMS was pretty straightforward, the Faraday principle, this one's even more straightforward. Basically, you take 2 electrodes, you put them on the scalp, you run a very low current between them on the order of 1 to 2 milliamps, and what happens is that creates sub-threshold stimulation in cortical neurons. And what I mean by that is below the point where they depolarize. It doesn't make them fire acutely. What it does is it incrementally changes their resting membrane potential to make them either a little bit more likely or a little bit less likely to fire over a given period of time. depending on which kind of electrode polarity that you're actually applying. So the received wisdom is that anodal stimulation is more excitatory, cathodal more inhibitory. And so the advantages from our perspective compared to TMS is that it's very safe. TMS has a small risk of seizures. No one has ever demonstrated any serious adverse effects. It's inexpensive. The most expensive units are about $10,000. The cheapest one is the one that you build when you go to Radio Shack with $100. And they're easy to use. And we are seeing, this is a result from an ongoing study, but we are seeing that you can actually change language ability in patients with aphasia using this kind of stimulation. So this is performance on the Western Aphasia Battery. And while these aren't night and day dramatic differences, like these guys are not trying out for the local theater company after they get brain stimulation, but they are significant and meaningful with respect to the amount of improvement that these patients experience and that they feel that they experience after having received stimulation. So just to decode that for you, so that's the Western Aphasia Battery, very common battery of aphasia severity, and you can see that the patients who are getting real stimulation are getting better, the patients with sham not 2 weeks, 2 months after stimulation. That's just a more flattering way of depicting the same data, showing that the degree of change in the individuals who are getting stimulation is much greater than the degree of change in patients who are getting sham. Before I quit, I'll just take a quick detour to say that we also are stimulating normal individuals and using this technology, both technologies really, to manipulate cognition and in some ways improve aspects of cognition. So this is from a recent study we published in which we used TDCS over the frontal lobe to manipulate aspects of creativity. So what we asked individuals to do here is to look at common objects and come up with novel uses for them, right? So you look at that big old clunky shoe, and you might say to yourself, well, if I was in a pinch and I needed to hammer a nail into my wall, I might use that as a hammer. It's got that crazy heel. All right, so it turns out that when we stimulate over the frontal lobe, right, individuals are more likely to come up with an alternative use to a common object. In other words, they don't fail to come up with the alternative use, and they're faster at it. Right? So we can enhance certain aspects of cognition, and this is just one of a number of examples, which I don't have time to go into, to illustrate that we can do this in normal individuals. But that sort of engenders a concern, right? So now you've got this cheap, easy technology that you can use to manipulate cognition in normal individuals, and so that's given rise to a do-it-yourself brain stimulation community. I mean, there are products that are being marketed, like that guy on the bottom left hand. That is being sold on the internet now to help you play your video games faster, right? The other ones are basically homemade units, right? As if you couldn't tell. And so this is my one plug to a room full of engineers about engineering. Like, we need better units because I don't want the future of brain stimulation to be left to, I don't know, that guy. All right, so in conclusion, brain reorganization drives neural repair, right? I hope I've convinced you of that with a couple of examples. Brain stimulation can facilitate plasticity, right? We talked about how the brain is capable of dramatic reorganization when it needs to be, but sometimes that reorganization is not optimal, and we can use focal brain stimulation to manipulate it in ways that improve recovery. Just something to watch out for, the emergence of easy, marketable brain stimulation I think that cosmetic stimulation is in the near future. And just, again, a word to engineers to build a better mousetrap, that we're always looking for better ways to more focally and more sensitively manipulate cortical function. All right, and with that, I'd like to thank the members of the Laboratory for Cognition and Neural Stimulation and you for your attention.
Thank you, Dr. Hamilton. Okay, next, we're going to have Dr. David Lyon. He's assistant professor and director of studies, anatomy and neurobiology at the University of California, Irvine.
Thank you. Well, it's great to be here just to see these two talks before mine. I'm glad to be a part of this. And also, as far as Long Beach credentials go, I gotta throw mine out here. So I'm actually a graduate of Cal State Long Beach 1992 from Department of Psychology, I got my bachelor's, and then I stayed on for a master's for a few more years. And I don't know the Latin for it, but my father also got his bachelor's and master's from here in the College of Engineering, which might seem like a good thing for me being here to speak to the College of Engineering, except I don't know anything about engineering. So none of it rubbed off on me, sadly. But I will try to say something relevant. Okay. Oh, yeah, here's me. Yes. So what I want to talk about is, it's shifting gears a little bit. I'm not going to say anything about the human brain. I'm going to talk about other brains, animal models that are used to study neuroscience. And what's nice about the animal models is you can get more invasive. You can do things that you're not allowed to do to your neighbors. And what I was going to say about the President Obama's Brain Initiative, a large focus of the initiative is actually on animal models, so it's not all just about studying the human brain. And I had a slide showing who the advisory panel was chosen, here we go, for, how'd you do that? Well, so to point out a couple of the core, the priorities of the Brain Initiative. So one is mapping the structure and components of circuits, and another then is manipulating the circuit activity. And so the kind of work I do is getting at these things, and you can do it at a finer detail in these animal models. Here's a couple of the calls for grant applications that directly relate to what I do. So developing novel tools to analyze cell-specific and certain circuit-specific processes in the brain. So getting at the fine details of the circuitry, and then being able to manipulate these circuits. Okay, this is the panel of experts, and what I wanted to highlight here is that the two co-chairs kind of span the whole range of the types of animal models that are important for neuroscience, coming from the worm to the monkeys. And I think they were chosen on purpose to kind of emphasize that a whole range of species are important to get at this question of how the brain works, how the brain is wired. So I use this to kind of validate what I do is important too. Just because it's not in humans, there's still a lot of useful information you can get. And actually, so I choose to study species more on the complex end too. And here are some examples. The kind of work I do is actually in visual cortex, and so I think there's a common theme going on here. And primates, non-human primates, are a really good model for visual system. They don't have it's not 41% of their cortex like the frontal lobe, but a good third of their cortex, to almost half, actually, in this macaque monkey, is all related to processing vision. So visual system is very large in these animal models, also in cat, and this close relative, they're a tree shrew. In mouse is very popular now for studying visual system, primarily because of transgenics. You can manipulate the gene expression of different neurons that allows you to target particular circuits for tracing the connectivity or for activating circuits, shutting off circuits. Mice are really emerging as a very key model in studying the brain. But for the visual system, it's kind of lacking in this higher level functioning and making it harder to translate to the human. And so what my lab's trying to do is develop sort of a workaround of this transgenics, and to use viruses to deliver genes to neurons. And so not in a sense of gene therapy, but this could also be an outcome of the kind of work we do. Here's a complicated slide. I'm just going to gloss over a few things, but, oh, wrong button. Darn. Let's see. Hopefully this works. That's dangerous. We use rabies virus, and it sounds a little bit dangerous, but they make sure we're vaccinated before we use this. But we've debilitated the virus. Okay, so we've genetically modified it and we've pseudotyped it. And what this allows us to do, of course, it's not working again. It allows us to control how rabies can act as a tracer. So I forgot to mention rabies virus, it's evolved to, oh, now I'm up over here. It's evolved to infect neurons. That's what it likes to do. And it spreads specifically through synaptic contacts. So it just doesn't diffuse through the brain and infect neurons. It goes through the synapses. And so it turns out to be a natural circuit tracer. Now I can't even get it. Okay, good. So here is a proof of principle example of how we use it as a circuit tracer. This neuron here, this is just in a brain slice in a dish. We've shot genes into this neuron, genes that allow us to use the virus as a tracer. A gene for this TVA receptor, which is the receptor for our now modified virus, and this rabies glycoprotein. Those are two key genes. And this DS red allows us to see which neurons have been shot. So we have our modified virus. Because this TVA receptor is expressing in that neuron there in the middle, the virus can infect that cell. And it won't infect other neurons. You can't see the other neurons because they're not expressing red fluorescent protein. But there are hundreds there. Then, because there's a rabies glycoprotein present, this allows the new virions that are generated in that neuron to spread to the neurons providing direct inputs. But it won't continue to spread. And so this virus causes expression of green fluorescent protein. That one neuron should express green fluorescent protein, and all the neurons connected to it should express protein. And so this is what you get. Now we've just labeled the whole circuit. All the inputs to that neuron locally have been labeled through this technique. And so now we're developing applications so that we can do this in live animals. That was done in a dish. And so the trick is you need to deliver these genes for the receptor and for the rabies glycoprotein. And the way you can do it is with other viruses. Because what do viruses want to do? They want to infect cells and deliver their DNA or their RNA so that more virus is made in those cells. But you can kind of hijack these viruses and insert your own DNA or RNA in there that you want to use for, in our case, for experimental purposes. So we've inserted in our TVA receptor and our rabies glycoprotein. Now there's a distinction. We have two different helper viruses, and we're using them to get at two different types of neurons. So in cortex, which seems to be the theme today, the two main types of neurons can be broken down into excitatory and inhibitory. So a single neuron's not going to be both. One is either going to suppress other neurons or one is going to excite other neurons. And we really don't know a lot about the circuitry related to the two different types because there's not a technique available to be able to trace the circuits of the two different types. And so that's part of what we're doing here. So we use these tricks. We use here this is a GAD1 promoter, but what that does is that will cause expression of our genes only in inhibitory neurons. So it takes advantage of the machinery in inhibitory neurons. This GAD1 promoter is activated in inhibitory neurons. Or this alpha-CAM-K2 promoter will activate, will make the proteins only in excitatory neurons. So now we have our receptor in either inhibitory or excitatory neurons, and we can trace the circuits. And so this was just pretty recently when we figured this out, and here's a test of it. Now all these cells expressing the receptor are just inhibitory neurons in the cortex. So they should be infected by our modified rabies virus, and these cells are cells infected by the rabies virus. There shouldn't be anything infected if the TVA receptor wasn't expressing. So this is it looks prettier here, the double labeling. You can see some yellow cells. This is where the initially infected cells are, and I highlighted them so it's easier to see. But then all these red-only cells, they're the cells that are providing inputs to these yellow cells. So this is all just locally, but throughout the brain, there are cells coming in to provide inputs to these inhibitory neurons. And we've worked out differences on these brain-wide inputs to inhibitory and excitatory neurons done in visual cortex in the mouse and found, in fact, that excitatory neurons are getting a lot more inputs from outside of their own area compared to inhibitory neurons. Inhibitory neurons seem to be controlled more locally, which was kind of an interesting finding. And we need engineers to do some modeling of this to help us understand what's going on. So I think there's a lot of opportunity for complicated models. Now, what we really want to do, though, is look at a more complex species, because in mice, you could already do this through transgenics, but this was just to test it out. So here now is in a cat. We like cats, so this is a surface view of cat's primary visual cortex. You can use intrinsic signal optical imaging to find a functional map known as the orientation preference map. So each of these color-coded regions correspond to large clusters of neurons that prefer a particular oriented stimulus. So all the orange regions prefer, say, a horizontal bar. When they're in different parts of our visual field, though. But these orange regions tend to be connected to each other. But it's not known if there's any sort of specificity of the connectivity with inhibitory neurons, and that's what we've looked at with our technique. So here are all the inputs to inhibitory neurons where that arrow is. And if you look in the color coding, we can see that these local inputs inside that circle, or these long-range inputs, they're tuned to the inhibitory neurons. And in my field, this was interesting finding because there's a debate on whether inhibitory neurons have any tuning and what's mediating this tuning. And now this is kind of getting even a little more vague, but you can generate these different tuning curves, or cells will have different tuning curves. So this is tuning curves from the same cell, but in one instance, the center excitation, this is more from the activity of this local population of connections. Whereas the surround suppression is more activity from those longer range connections. And you can see that the tuning, these numbers that I've circled here, like 0.71 kind of matches with 0.78. But what you can start doing is correlating the tuning of the inputs to the function of the cell. And so this is just kind of on an anatomical level. But what we really want to do is start manipulating the function of these neurons to see how these circuits cause a neuron to fire and what basically how the visual system is working. And so you may have heard of this method called optogenetics, where you can deliver a membrane channel, a gene for a membrane channel to a cell. This will get embedded in the membrane, and if you zap that cell with blue laser light, it causes, if it's a neuron, it causes it to spike. And so in this way, you can manipulate the circuits. And so a colleague of mine from the Salk Institute, he developed this easy way to kind of insert your own genes into rabies virus. And so now you can do it with this gene for channel rhodopsin, which is this light-gated ion channel. So anyway, this is just showing that you can activate the cell with light by delivering this gene from a virus. So this is done, I think, also in a mouse. Mice are easier to work with in a lot of ways. All right, but what we want to do then is we can record from this initial region there, activate this cluster with light, and see what effect it has on here. And this might seem like a real basic thing to do, but this is a big step forward in getting at this kind of fine-scale circuitry, and especially in a non-rodent model. Okay, and what we want to do even further is kind of get at these different inhibitory subtypes. So there are inhibitory neurons and there are excitatory neurons. But inhibitory neurons are very diverse. And this is an example of cross-section from mouse visual cortex, and different inhibitory neurons are labeled here. And you can activate these, you can target them in different transgenic mouse lines, but you can't do it in a monkey or a cat. So what we're trying to do with our helper viruses is to be able to target these inhibitory subtypes with specific promoters, and so that's what's shown there. So kind of a window into new methodologies coming out in kind of the neurobiology side of things. And so here are some of the people working in the lab. Youngjun there basically did all the work. Hanjun is our new virus expert. And then I just want to point out this guy, Moritz, came free from Europe as an exchange graduate student and did a ton of work and really made me appreciate free graduate students. Yes. And my collaborators from the Salk Institute and Allen Brain Institute down there. Okay, that's it. Thank you.
Thank you, David. It's really interesting, if people want to get ready to ask questions. I think one of the things that I heard, and I can really see it, is some of the simulations, the modeling programs. I think that's going to be a huge thing that we can go and, from an engineering standpoint. But just to lead it off, I do want to ask the question, and I'm going to come up there in a second, but I want to get the question out, is with all this new money coming in there, the $100 million, and I don't know how much you guys know about it or be able to use it, is there, are you going to be able to take on more students, do more modeling, do new equipment with that? Do you think it's really going to be a big help in the near term? Or you think it's really something longer term, or do you even know about the funding?
Do you want the PC answer or
Whichever you want to do.
I think that the best part about the BRAIN initiative is the awareness generated from it. The awareness, the public awareness of neuroscience research, I think that's really what's helped. $100 million is nice, but that's going to fund 50 labs or something, and there are a lot of labs out there, and it's probably going to fund the labs that already have too much money to to spend right now. But I think it's to get things started. The $100 million, probably you could squeeze it through Congress, and get things going. So I think it's only going to get better.
Any other comments on that?
Yeah, I mean, I agree. In addition to just raising awareness, I think that, well, I should say, in addition to raising awareness with the public, I think that it, at least in my institution, has served as a bit of a catalyst for people from different disciplines to realize that they could work together towards this common aim. And I really think that's its value, again, just to echo David, more than the amount of money, because that amount of money spread over the number of investigators we have in the country is not all that much, but it has started, at least with some of the people I'm working with on our proposal for the initiative, again, sparked some very novel thinking about what the next generation of imaging in the brain should be. And even if we don't get funded for that, that's going to fuel us to other sources and other ways that we will get this work done.
Thank you. Tülin Manger, Electrical Engineering Department. When I listened to you, Dr. Lyon, and then Dr. Hamilton, your long-range and short-range impacts or kind of working to collaborations could probably explain some of the plasticity we see, how different parts of the brain are being recruited. I'm not sure if that's an accurate statement, and I was wondering if both of you could comment on that. If I understand what you described correctly, is that your future of your work could probably do a lot more in explaining how we get this plasticity and other parts of the brain become more experienced, so to speak, in different functions that they weren't before?
Sure. So let me say at the outset that I think that I need the work of people like David more than he needs me, but let me just say that I completely agree with you that one of the shortcomings of the kind of work that has been done in brain stimulation to date has been, in my opinion, a failure to link sort of systems level and above plasticity and communication in the brain to the mechanisms that underlie them on a cellular and molecular basis, right? And so the work in that area, I have to say, has been to date pretty siloed. And obviously, those are silos that are going to have to be broken down if we're going to come to some more overarching or comprehensive understanding of functional brain architecture, right? And so that's the reason why I say I think I need David's kind of work more than he might need mine, because on some level that's sort of a speed limit, an epistemological limit to the kinds of work that brain stimulation is capable of until we understand some of the mechanisms that actually cause cells to communicate and to strengthen and modify their connections.
So, I mean, as far as short range and long range related to plasticity, I think they're interrelated for sure. Short range is going to have a lot more of a inhibitory effect than the long range in some ways. Also, short range just has more of an impact in general. It's kind of where most of the connectivity in cortex anyway, we see something like 85% of the inputs to the cells are all coming from right next to the cell, and the 15% is coming from everywhere else. So short range, I think, has a big impact just on the function of the cell. Plasticity kind of depends on, are we just talking about normal development, or are we talking about recovery from injury? What's injured, I think it all plays a role, and rehabilitation and recovery is all about kind of whatever circuits are left, getting them to take on more of the workload, and maybe even forming new connections or making more of the connections that are there. So I think there's still a lot to be figured out for that.
Hi, Walter Martinez, College of Engineering, and Cal State Long Beach graduate twice as well. So I just have a quick comment and a question. One is, I just read on Scientific American that they have opened the head of a mouse and they injected a special dye in the brain, and they could actually physically see the activity of the brain as the signals were activated and the dye turned into a different color in the brain. So I don't know if you heard of that, but that's one of the comments. But my question is, have any of you done any work with autistic people and noticed the signals as to how different are they from people who are not autistic? And can we do something with those signals and help this kind of people?
Yeah, I actually used an autistic mouse model that were for a model of Rett syndrome, which is kind of a more severe form of autism. But what we're just trying to do some basic circuit analysis to see if the density of the connections is different sort of across the brain areas. So far we found that it looks to be pretty similar. And so it may not be in terms of number of connections are similar. So it's probably something else, an imbalance in the way the neurons respond to the connectivity.
Did you want to answer?
No.
Thank you. My question is directed to Dr. Lyon. You mentioned using rabies virus to map the interconnections of neurons through the synapses. But then you mentioned you can do it for inhibitory as well as excitatory. So, for example, a particular virus can only propagate through inhibitory neurons. Can you explain a little more how that's done?
Yeah, and I should clarify. So it starts at one of those neuron types. So it'll start in inhibitory neurons, but but the connections that we label are all the connections coming in. So we're labeling the excitatory and the inhibitory, which is actually good because the inhibitory connections are only right there. The inhibitory inputs, they're all, in cortex, inhibitory projections are all very local. But the excitatory ones are connecting across the brain. So you have kind of this local processing, the local excitation and inhibition. Signal goes out as excitatory, and it will kind of start this whole process in the next area. So we're not, I guess we're lucky in the sense that we don't care if we're just labeling the inhibitory inputs coming in, because there's not a way to do that yet, for sure.
My name is Tim Jornides, and I have two questions that can be addressed to any one of the three of you. The first is the imaging of the brain. Some psychiatrists have made a fairly big deal out of it in treating bipolar disorders and depression. I wanted to find out what is the most recent evidence that has come out of research indicating that, yes, you can do successful imaging and perhaps predict or treat or help the recovery from those disorders. The second question is there anything on the horizon about electronic devices that may be implanted somewhere in the brain with the goal in mind to, say, allow blind people to see a little bit, crippled people to have some tactile capability, and those types of things. Thank you.
So with respect to, those are two very different questions. So I'll try to address each of them a little bit. So there is some evidence that there are quantifiable differences in imaging, to some extent, both anatomical and functional imaging, between individuals with depression and individuals who are not depressed. The practical ramifications of those changes in brain activity, I think, are still yet to be fully borne out. So I'll give you one example. There is one avenue of research or sort of group of researchers that has found that there's difference in activity in a particular part of the sort of limbic system. I guess the specific names aren't all that important, but sort of near the underside of the frontal lobe and something called the anterior cingulate. And there's been some work using neurostimulation to try and put stimulators in that region of the brain to see if we can help these individuals out. And that has borne sort of mixed fruit. It's not clear that that's going to pan out. On the other hand, there's some evidence that there's also differences in activity of frontal lobes in these individuals compared to otherwise healthy individuals with respect to mood. And there's actually been work in brain stimulation, in particular TMS, and that does seem to have borne some fruit. The transcranial magnetic stimulation now has an FDA approval. It got approved in 2008 as an indication for the treatment of depression. It has sort of narrow indication. You have to have failed one medication, but not two. It's a sort of a weird thing. But the point is, your doctors can use it to treat depression now. And that was sort of born originally out of observations of differences in brain activity. And then your second question, oh yeah, so there is work certainly being done looking at a variety of different sensory prostheses, sort of whether or not you can sort of replicate the function of the retina. Certainly, I think a more advanced or an area that more people are familiar with is cochlear implants, right, in terms of replicating sensory processing. So, there is work, and there are also systems that transduce from one sensory modality to another, right? So for example, for blind individuals, there are actually sensory systems that transduce visual information into sort of tactile impressions that they can feel and then sort of make a mental image out of what they're seeing out of those tactile impressions. So there's a variety of different avenues of research, avenues of work that are being done, either to leverage the other sensory abilities of individuals who've had some form of sensory loss, or in engineering to try and reproduce those sensory modalities.
I want to thank all of you for sharing your information and enlightening us as it relates to the brain. Dr. Hamilton, I wanted to focus on you specifically because you kind of demonstrated that the brain has the capacity to transform and adapt if it's been injured, to use another part of the brain to reciprocate for a function that it used to do. I'm curious, because you sort of sent out an RFP for some additional stimulation machines through the engineering students here, if over time, let's say that the brain has adapted and is using another part of the brain, while that part is being used for that function, if the other part that's been damaged can be stimulated by the RFP that you issued to repair what's been damaged on the original site for it to come back strong. It's sort of like, if you break your leg, you don't put pressure on it, the other leg takes the weight until you can then bear weight on the other side. Do you think that in the future that's a possibility? Because you demonstrated also with people who didn't have, they had a part of the brain that was dormant, that wasn't being used for the Braille because they didn't need it, that it strengthened over time and they were able to use that part of the brain for that function.
So to address that question, I'm going to glom onto your metaphor of the broken leg. And I'm going to apply that metaphor to a phenomenon that we see in language recovery. So there was a lot that I couldn't tell you in the time span that I was given about how the brain changes its representation of language in the setting of aphasia. So one of the things that we didn't have time to discuss was that there are temporal differences as well. So if you were to image the brain and its functional activity with respect to the performance of language tasks in many of these aphasic patients, right after they had their stroke, that pattern of functional activity would look very different than a couple of weeks after they had their stroke, and that would look different than a couple of years after they had their stroke. And one thing that seems to be the case is that there appears to be more of a shift into the right hemisphere, into the intact hemisphere, in that subacute phase. Think of it as you broke the leg, and so now you're limping on the other one, right? However, in the chronic phase, it seems like there is some recovery. There's some shift back, at least in some imaging studies, to some extent, back to the left hemisphere, so that you're talking about more of a bilateral network. Right? And so you can imagine a couple of things here. You can imagine stimulation systems that do something different, because, just think about it. There's no necessary reason why the kind of plasticity and the kind of compensation that you need to induce in an intact system that was doing something before, that now needs to acquire a new function, right, should be exactly the same as the process you would need to stimulate recovery of a system that's been injured that now needs to reacquire some network function, right? So you could imagine that you would need, that in a future of brain stimulation, that you would have stimulators that were so specific to the kind of plastic process you need that it would be able to differentiate between the kind of stimulation you need to give to facilitate the acquisition of new function in intact brain versus the kind of stimulation you would need to rehabilitate lost function in perilesional regions of brain. Certainly, like, that would be one dimension where you can imagine engineering yielding specificity with regards to the effects of stimulation.
Okay, we have time for one last question.
Let me just do a follow-up.
Go ahead.
I'd like to ask Dr. Mendez, do you see, with all the injuries you see, with the VAs, the veterans, do you ever see, recovery from where they may be using different parts of the brain? Do you ever see that type of activity, with some of the patients that come in? I'm just curious about that, because I think that's such an important area, you know, when we really have people with serious injuries. If
You woke
really
me up
see
from
that.
my nap.
Yeah.
No, actually, fortunately, yes, there's a lot of wonderful rehabilitation that's going on, and it is using compensatory techniques, shifting function to other types of cognitive and physical activities, and so there is it's kind of a different form of plasticity, if you will. We think of it as a compensatory process of taking, you know, maybe the right hemisphere taking over what the left hemisphere was doing, and so on. So the VA has a wonderful rehabilitation program for the veterans. Could always be better. So write your congressperson.
Could I just comment also on the metaphor of the broken leg, waiting for the other leg to heal and the good leg compensating? Really what you have in recovery is whatever is left compensates, and the broken leg's not going to come back. The bones aren't going to come back for the most part. I mean, there might be some damaged neurons that can recover, but a lot of the neurons are gone, and you don't regrow neurons in an adult brain. And this is where stem cells come into play. So at UC Irvine, they're doing a lot of spinal cord research to get the motor neurons to grow back, and so they're using stem cells to do this. And so this will be a way that the leg can recover, hopefully.
Okay, we're going to end with a student question here.
Yeah, hi, my name is Joey Seamus. I'm an undergrad student here. And my question is to Dr. Hamilton about the last portion. The last portion of your talk was about the electron, or the electric, I guess, stimulation of the brain that helped people become more creative or somehow increase their performance. I was just wondering what you, I guess, personally thought, or if you could expand on that, like about the legitimacy of that, or like, I don't know, because I thought that was really, really phenomenally interesting, like that you could, I guess, theoretically become maybe more creative or smarter from it. So I don't know, that kind of blew me away. So I just would like to hear a little more. Thank you.
First, let me ask you, can you operationally define legitimacy? Otherwise, I could be talking here all night.
Okay, that's a fair question. Well, I don't know, because I mean, I guess from the study, it did make people more creative, right? So do you think that is a viable? I don't know. I guess it increases performance, but do you see it as being something that's actually helpful to people, or is that kind of like a fad or something? I don't know.
I'm going to break your question down into two categories. And so the two categories are, does it work, and should we do it? All right? Okay. And so the answer to does it work is probably a little bit easier to answer. You know, it's certainly the case that we can induce temporary improvements in aspects of cognition. You know, it's done by lots of researchers in this area. We do it in our laboratory. We've done it in a number of domains, paired usually with some type of activity that's meant to tap into that particular mental operation or cognitive task. Whether or not it works in the long run, right, there's actually a dearth of evidence because most of the work in this area has been done in cognitive neuroscience, where the point is to establish the relationships between brain areas and brain networks and cognitive operations. We're trying to find out more about the brain. They're not treatment studies for normal people, right? So there's actually very little work where individuals who are normal have come in for weeks at a time, gotten their brain stimulated, and then figured out whether or not it made them, you know, supermen or women. There's less information in that regard. However, we have reason to suspect from our patient studies, where we stimulate patients a bunch of times and they seem to get better and that seems to last, that it might in fact be the case. Now, the question of should we do it, that's much more complicated, right? Because then you get into all sorts of things. Well, first of all, what don't we know about what brain stimulation is doing to brains, right? Think about cognitive neuroscience. You stimulate a brain. Think about the way experiments are set up. You stimulate an area of the brain, and you think that area of the brain is involved in some type of mental activity. So what do? You test that mental operation to see if it got better. Well, it turns out brains are pretty complicated, and one brain area is connected to a bunch of other brain areas, and one brain area doesn't just do one thing. Well, you didn't test everything the brain does, so what's the effect on all the other stuff, right? So we don't know what the long-term ramifications are of stimulating a normal person over and over again in one site to improve one thing. Secondly, then you get into all sorts of complicated, like philosophically complicated questions. If you can alter core properties of your cognition, at which properties, at which point do you say, well, I've altered something essential about myself? Like, I'm a different person than when I started doing this brain stimulation, right? And that's a complicated question too. And then you get into things like distributive justice, like who gets to get stimulated and who doesn't, and all sorts of things. Like coercion, like should people in the military be stimulated so they can detect things faster, and should they be forced to be if they don't want to be? Like it just gets really murky, right? I've actually written a little bit about the ethics of this and how murky it can be. I think at the end of the day, we might need some form of either or education and or to some extent regulation, just in terms of safety of what's being built out there. I mean, there are people putting car batteries, with sponges on their heads, right? So we might see some level of oversight and some level of education from the scientific community about what is safe, what seems reasonable, and what you couldn't expect from brain stimulation.
Thank you.
Just add one comment. In our frontotemporal dementia work, we've done a lot of things with art. And when it's left hemisphere predominant, left frontal predominant, it often releases a lot of artistic ability. And this is usually in the visual domain, but sometimes it's been in the auditory domain, where people who never did very much, and it's more than just, most of it is just representational art, but some of it has been quite abstract. So sometimes when you suppress one cognitive function with disease, you get a release of creative abilities. In this case, we think it's suppression of language then releases a lot of visual artistic ability. Just a thought.
Okay, I'd like everybody to really thank the speakers again. And just excellent. Thank you very much. And our next lecture is going to be October 22nd. Don't leave yet. October 22nd. We're going to be looking at doing a couple different, I talked about last time, we were going to do 2 in a series, and the next 2 will be along the topics of aerospace and whether we do satellites, airplanes. We're still working out the topics, but we're going to try and get some real good topics, a couple different topics for the next 2 lectures. And I'd like to give this back to Dean Ghoshani. Thank you, Nick.
Thank you, distinguished speakers. Thank you. It was stimulating, thought-provoking, with a nice mix of humor, and thank you. So, and my apologies for technical difficulties we had, and hopefully we'll fix it for the next one. I would like to thank the staff who have worked on putting this together. It's quite a bit of work, as you can imagine. Our development office, Nicole Forrest Boggs and Nikki Algren Cheveria, thank you very much. Please make a note of October 22nd for our next lecture. Thank you for your participation. And again, I'm grateful to the speakers. There are some gifts that we will be presenting to you, and I think that concludes our, down here? Okay, thank you.